Hello guys, welcome to Dr. Science. So today we are going to talk about what are toll-like receptors. This is my part 2 explanation regarding the innate and adaptive immunity. So toll-like receptors comes under innate immunity. So before going into what are toll-like receptors, I will give a brief explanation of toll-like receptors. Okay. So suppose this is a cell, and this size cell is called as dendritic cell. Okay. Dendritic cells are a type of innate immunity cells, as I told you previously. Okay, and these dendritic cells have certain receptors present on it. Suppose we draw another cell here, and this cell is coronavirus, and this coronavirus having certain patterns like this is red pattern, this is green pattern, and this is blue pattern. These dendritic cells will attract those patterns, it means attack those patterns. Okay, hence these receptors here are called as pattern recognition receptors. Okay, or PRRS, pattern recognition receptors. So what are these green, red and blue surface antigens here? These are called as pathogen associated molecular patterns. Okay, these are called as pathogen associated molecular patterns or PAMPs. Okay, and these PAMPs are exclusive, not only exclusive to coronavirus, they are also found in certain viruses like influenza virus. Okay, influenza virus also have these type of patterns present on it like red, green and blue, and these are helpful in infection, causing infection. Okay, okay, so pattern recognition receptors or PRRs. Okay, PRRS, which are present on dendritic cells or any of the innate immunity cells. There are mainly three types or four types. Okay, one is toll-like receptors, the second one is nod like receptors, the third one is C type lectin receptors, the fourth one is rig rig like receptors. Okay, so these are the four types of receptors. Okay, I will discuss each one of them. So suppose this is the plasma membrane here, this is the plasma membrane, and everything outside or everything which is present extracellular is called as extracellular toll like receptors. Okay, not only that, C type lectin, this is C type lectin receptors are also present extracellularly. So what is present inside the cell? In the cytoplasm, suppose this is the free floating cytoplasm. In the cytoplasm there are freely floating receptors. Those are nod like receptors. Okay, in the cytoplasm we will find nod like receptors. So intracellularly there are vesicles. Okay, these vesicles are called as endosomal vesicles, and these endosomal vesicles contain receptors present on it. These are also toll like receptors. So toll like receptors are present extracellular and also intracellular. Intracellular toll like receptors are present on endosomal vesicles. Okay, and C type lectin are present outside. Okay, now we will talk toll-like receptors. Okay, toll-like receptors are first found in Drosophila. Drosophila is a fly. Okay, in the fly they found these toll-like receptors uh, which are helpful in recognition of microbes. So what is the function of toll-like receptor? Suppose this is the plasma membrane, this is the toll-like receptors, and one microbe visited these toll-like receptors and they got activated. Okay, once they get activated, they have mainly two functions. They will first they will activate the transcription factors, which will eventually activate two of the main important pathways, which is Nfkb pathway. What is the function of Nfkb pathway? Nfkb pathway increases the cytokines. Okay, cytokines will attract more of the leukocytes. On the top of that, they will increase the cell adhesion molecules. Okay, so once these cell adhesion molecules are increased, they help in adhesion of leukocytes. Another function is that it will increase the production of interferon. Interferon regulatory factors. Okay, what these interferon regulatory factors or IRFs do? These IRFs will eventually increase the production of type 1 interferon. Okay, type 1 interferon is a type of antiviral cytokine. Why it is known as antiviral cytokine? I'll just explain to you. It is known as antiviral cytokine. What does this mean? It means the interferon will convert the active it will convert the active viral genome into inactive viral genome. Suppose if there is any viral RNA, it will convert this active viral RNA into inactive ones. Hence, the type 1 interferon is called as the antiviral cytokine. So these were the functions of toll-like receptors. One is increase in the production of Nfkb pathway and increase in interferon. Okay. Next we will talk about node-like receptors. In node-like receptors. So node-like receptors are present in they are present in cytoplasm. So what happens to these node-like receptors? So node-like receptor is a multimeric protein. Okay, so this is one protein, this is one protein, this is another protein. So this entire protein complex is called as the node-like receptor. Okay, or NLRS, node-like receptors. So this is the sensor for it, and again here is another protein. This is called as the adapter protein, and finally there is an enzyme attached to it. This enzyme is caspase 1. So whenever node-like receptors are triggered, these multimeric complex get attached, and this caspase 1 converts inactive interleukin 1 into active interleukin 1. So what is the function of interleukin 1? The major functions of interleukin 1 is, first one is fever, the second one is inflammation. Okay, so interleukin 1 causes fever and inflammation. There is a disease called as syndromes called as auto inflammatory syndromes. Okay, what happens in these auto inflammatory syndromes? In auto inflammatory syndromes, the node like receptors are mutated. What happens if the node like receptors are mutated? There will be increased conversion of inactive interleukin 1 into active interleukin 1, and this will eventually cause fever and inflammation. So what are the trigger factors which causes the activation of node like receptors? Node like receptors are activated mainly by crystals. Can you name some diseases which there are where you found so many crystals? Yeah, in gout. In gout, you found these urate crystals. These urate crystals will eventually increase the activity of node-like receptors, and there will be increased conversion of inactive interleukin 1 into active interleukin 1, and there will be fever and inflammation in joints. Okay, and the second one is obesity related type 2 diabetes. Okay, where there is increased cholesterol crystals. These cholesterol crystals will activate crystals. These cholesterol crystals will activate the node-like receptors, and the mechanism already. Okay. So next thing is we will talk about C-type lectin receptors. C-type lectin. So these C-type lectin receptors are nothing but they are present mainly extracellular, right? C-type lectin are present extracellular, and these extracellular C-type lectin, what they will do? They are only activated by fungal glycans. Okay, so fungus have these glycans present on their surface. So C-type lectins are only activated by glycans present on the fungals. The C-type lectin are most abundantly present on, the first one is dendritic cells, the second one are macrophages. These C-type lectins are mostly abundantly present on dendritic cells and macrophages. The last but not the least are the rig-like receptors. Okay, the rig-like receptors are present in the cytoplasm. Okay, what they will do? The rig-like receptors, they will convert the active viral RNA into inactive one. So that was a brief explanation. Now we will go to the images. Okay, so these are the toll-like receptors. As you can see, this one is extracellular toll-like receptors, and they are activated by bacterial products. And these are C-type lectin, which are mainly present on macrophages and dendritic cells. And here the microbial polysaccharide is glycans. What glycans? They are fungal glycans. Okay, so here are the vesicle. I told you the endosomal vesicle here, and these endosomal vesicles which have toll-like receptors present on their surface are called as endosomal toll-like receptors. Next, they are cytosolic toll-like receptors, which are called as nod-like receptors, which are mainly activated by bacterial and peptidoglycans. Not only that, they are activated by, yeah, you are right, urate and cholesterol crystals. Okay, and here is rig-like receptors, which will convert the active viral RNA into inactive viral RNA. Okay. Okay, here is the nod-like receptors. As you can see, nod-like receptor is a multimeric protein complex, right? Nod-like receptor is a multimeric protein complex. It consists of sensor. So this is the sensor here, this is the adapter, and it contains an inactive enzyme called as caspase 1. So whenever nod-like receptors are activated by, yeah, potassium influx crystals such as urate or cholesterol, bacterial products, and reactive oxygen species, the caspase 1 will convert the interleukin 1 here. As you can see, the pro interleukin 1 getting converted into active interleukin 1 in the presence of caspase 1. So caspase 1 is the enzyme which converts the inactive interleukin 1 into active interleukin 1, and this interleukin 1 causes acute inflammation. Okay, so that was about toll-like receptors, and please wait for the adaptive immunity part in part 3. Thank you.